Awesome. So thank you so much, everyone, for coming along today. I really appreciate seeing people in person. That's really cool. So my name is Chloe Yap. I'm a visiting student in Mike Gandal's lab at UCLA. And I come from the University of Queensland, from the land down under. And today my presentation is titled Autism-Related Dietary Preferences Mediate Autism Gut Microbiome Associations. My best party trick is a good Australian accent, so let's see how that goes. Oops. Sorry, team. There we go. So to start with, what is autism? Autism is a neurodevelopmental condition that is diagnosed on the basis of social and communication difficulties and repetitive and restricted traits. There's been increasing interest in this association between autism and the gut microbiome. And that might not sound like the most intuitive association right off the bat. And so I'll give a bit of history into how we got to this point. So it's been well known for quite some time that gut conditions are more common among kids on the spectrum. And by gut conditions, I mean like constipation, diarrhea, abdominal pain, and so on. In the past 10 years or so, there's been this huge surge of interest in microbiome research, the trillions of bacteria that live on us and within us. And so I guess somewhere along the line, people put 2 and 2 together and thought, I mean, maybe if gut conditions are more common among autistic kids, then perhaps their gut microbiome is different as well. There may be associations, maybe there's even causality. And so that prompted some initial human studies, some of which found some associations between specific bacteria and autism, but overall those were quite inconsistent and they were also small and underpowered. This, though, did progress to animal studies, in which there were kind of experimental changes to the microbiome in rodents, and then that was correlated with behavioral changes in these animals that seemed to mimic autistic traits in some way. And then more recently, there has even been progression to phase 1 fecal microbiota transplants, or more colloquially called transfusions. And this is actually an important sort of question at this point, because there have been more and more sort of industry players marketing therapies that claim to modify autistic traits by modifying the microbiome. And so these are being marketed to parents, and parents are obviously concerned about doing the best for their child. They are asking questions of clinicians, what is the best thing to do here in the absence of good evidence? And so far, though, I think it's safe to say that the field has been hampered by studies which are small in size, inadequate correction for multiple testing and confounders. And in fact, the field stands as having more reviews than primary articles, which is a bit of an interesting statistic. These are just some of the, things that you can find on the internet claiming to sort of have these microbiome quote unquote therapies for autistic kids. You have fecal microbiota transplants, you have these dietary therapies, probiotics, and supplements. And can I also just say that these are costly? Sometimes parents have to reorient their whole life around these diets. And for some of them, you don't know what the harms or what the side effect profiles are. And so we try to answer or provide some clarity to families and parents and clinicians by performing our own study that was large and kind of robustly and well designed. And we did this leveraging the Australian Autism Biobank, which is this national resource for studying the biological basis of autism. We had data collection from 4 of the biggest cities in Australia for a bit of geography. And recruitment ran from 2015 to mid-2018 and recruited a total of around 2,500 participants. So one of the cool parts of the Australian Autism Biobank is that it unites really deep phenotypic data with kind of quite wide-ranging genomic data. So just quickly to talk about the phenotypic data, we have a host of different measures of the autism spectrum in the autism group, as well as in kids without a diagnosis. So they're siblings and unrelated undiagnosed kids. All participants were offered an IQ test. That's important because sort of developmental delay and autism are sometimes or often associated, as well as family questionnaires, medical history, co-occurring conditions, and really importantly for microbiome studies, dietary data and stool consistency. And then we also, just to complete the picture, had data from their parents as well. And so that's the phenotypic data. We've also got a lot of genomic data. We've got genome-wide SNP data from pretty much everyone. DNA methylation in around 450, plasma lipidomics in 750, and then of course the gut microbiome data, which I'll be focusing on today. So let's get into the guts of it. Let's talk about the study itself. So in this study, we performed the largest autism stool microbiome study to date, leveraging this Australian Autism Biobank plus this Queensland twin adolescent brain cohort. And one of the kind of important features of this study is that we had really deep phenotyping data, as I've described before. And in terms of the metagenomics data, this microbiome data, we worked with Microba, which is a company who have a very large microbial database and profiler, which sort of exceeds what is available in the public domain. This is a schematic of our study design. So we've got this sibling comparison of 50 pairs nested within a case control comparison of 100 per group. And that's important because the microbiome has strong environmental effects, of course, and so the family data helps to control for that. And then we've also, then this is just a schematic of the group demographics. The age ranges from 2 to 17, so kind of quite the pediatric range. Overall age does match pretty well between groups. There is unfortunately a bias towards males, but that's expected given the epidemiology of autism, which is more likely to be diagnosed in boys. So now I'll talk about the input data sets and QC. And first I'll talk about the metagenomics data. Metagenomics data is basically whole genome sequencing of poo samples. And because it's whole genome sequencing, you can get high resolution taxonomic information, so to the species level of bacteria, viruses, whatever you choose. And then you can also have functional data because then you can find the genes that are encoded by each of these species. And because this is a QCBio seminar, I can only hope that some of you are interested in sort of microbiome data analysis in general. And there's a couple of quirks here for those who I guess haven't worked with this sort of data before. There are two sort of major challenges. Firstly, microbiome data is compositional data. It's sort of this ecology. And so each sort of species is just one part. It's a composition, it's a proportion. And so that's kind of a problem analytically for a couple of reasons. Firstly, it means that if you have greater sequencing depth for those samples, you're more likely to pick up rarer species. And so that could drive some like false associations. And the way that we dealt with it, and I think probably at this stage most people do, is to perform rarefaction. So essentially you might choose 7 million reads as a threshold, sample 7 million from each person, and then unfortunately you kind of throw the rest away, but that's just how this is often dealt with. The second issue with compositional data is that it violates independence by adding up to 100%. You know, if one goes up, the others go down, and it's hard to kind of figure out, you know, which is actually going up or down. And so one way to get around this is to perform a log ratio transformation, which was proposed by Atchison. And the specific flavor that we chose was the centered log ratio transformation. Oh, that's an RE instead of an ER, my bad. Where basically you interpret everything relative to the geometric mean, and then you interpret that accordingly. The second challenge of microbiome data analysis is that it's very zero inflated sparse data. So there are some bacteria which are present in like one person. And so how do you deal with that? The challenge with that is interpretation. In terms of interpretation, it's kind of unclear whether it's biological, does it actually represent something, or whether it's just because you didn't sample deep enough in terms of read depth. But the other issue, which is probably a bit easier, I suppose, to get around is the question of model choice. And so a few ways, which I guess at least we dealt with it, is to sort of approximate mixture models, so rare versus common, bacterial or species or whatever you choose. Or you can also do a two-part model where you sort of binarize data, test for presence or absence. And then for data where it's kind of a bit more normally distributed, then you can deal with that in the usual fashion. And the reason why I went into that detail is because that's what we did for our QC. So removing these really rare ones because they're sort of hard to deal with for some of our analyses, doing this transformation and kind of doing these splits of common versus rare. So then the other sort of class of data we dealt with was the phenotypic data. And we had dietary data from the Australian Eating Survey. I'll talk more about that on the next slide because that's quite important. Stool consistency, which is probably, the Bristol stool chart is probably one of my favorite clinical scales ever. It's literally like a chart of poo and like food descriptions. And then psychometric and demographic data that I mentioned earlier. So, sorry, still pestering you with like the nitty gritty stuff, but I guess it's kind of important here. Just to go a bit more into the dietary data. we looked at it in two ways. So it comes in its raw form as a food frequency questionnaire of 120 food items. So we dealt with it that way. The other way is that it sort of, the algorithm sort of spits out derived variables. And one interesting derived variable was for each person sort of splitting up their proportion of energy of food intake that is provided by each of these food groups. So we have healthy or core food groups like fruit and veg, and then non-core or unhealthy food groups. And then we ran a principal component analysis based on this with compositional sort of aware methods. And then what these can then be interpreted as based on the loadings on the principal component is dietary profiles. So principal component 1 represents high plant-based sort of diet, principal component 2 is high in dairy, and principal component 3 is high in meats. Cool, so there's a lot of different ways in which one could test for an association between autism and the microbiome. And so I'll go through all of these, but first let's start with the question, is there a relationship between traits and the microbiome as a whole? And the way in which we quantified that was to ask what proportion of variance is associated with or explained by the microbiome. And this can be interpreted as representing the upper limit of predictability under an additive model. And the way in which we did this is to build an omics relatedness matrix, where basically what you do is for each pair of people, quantify how similar they are based on a given data set. So for example, microbiome species data, as shown here, rare and common, based on genes, rare or common. And then we also, just for funsies, did a dietary one as well. And then you can then calculate the variance in these traits, for example, age, autism, stool consistency, and so on. You can estimate the variance of those traits associated with this matrix using OREML, which if you're familiar with sort of the quantitative genetics literature is like GREML for omics data, which is basically a mixed linear model approach. And so now to the results. We took age as a benchmarking trait because others, Rothschild et al. 2021 bioRxiv, did a similar analysis quantifying the proportion of variance in age explained by the microbiome in a much larger data sets of adults. And they found that was about 23%. That's this blue line here. And our result at the species level was very consistent. So that was nice to see. But interestingly, when we looked at the genes that are within this microbiome, they explain almost all the variation in age. So that's interesting. And then we turned to autism diagnosis. And in stark contrast, there was negligible association between the microbiome as a whole and autism diagnosis at the species or the gene level. But interestingly, there was an association with diet, which is kind of our first clue that maybe there's something interesting dietary going on here. And then we also looked at stool consistency and diet, because these are traits in which you would intuitively expect a relationship with the microbiome. And we did find some strong relationships there. There's some other observations which aren't shown on this plot. We also looked at some other neurodevelopmental traits, so like IQ and sleep problems. Again, basically negligible variance explained. And then we also tested kind of putting multiple of these relatedness matrices into the model. And basically it was interesting to see that adding taxonomic data to functional data, for example, explained more variance, which suggests that they contain slightly different information. Okay, so that's a cross for our first question. Is there an overall contribution? And so the next question to ask is, are there individual species associated with autism diagnosis? And we tested for this using ANCOM, which is a non-parametric approach, which accounts for compositionality and is also good because it controls for false positive rates well. And in this analysis, we only found one bacteria species associated with autism diagnosis. When we did a differential presence testing, so binarizing the data, this was also the top hit, although this was a nominal association. But I think the takeaway of this is that there was little evidence of individual species being associated with the autism microbiome, and we failed to replicate the previous smaller studies findings that have previously been reported. So we also have this functional data, and I mean, I guess the trade-off here is that there's over 2 million genes and 250 people, so multiple testing is going to kill you. And so to get around this, we basically limited our gene level analysis to just the genes that are directly encoded within the genome of this bacteria, which was a hit, hypothesizing based on your prior that you're going to find more associations. And we did find some associations, and these genes corresponded to sort of metabolic processes like amino acids, purines and pyrimidines, carbohydrates, and some other sort of bacterial processes. Another way in which autism, or in which there could be associations between a trait and the microbiome, is on the basis of species diversity. There's two ways in which to measure diversity. There's alpha diversity, which in its most basic form basically counts how many different bacteria are there within an individual. And then you can extend that to other things like Shannon index, which account for evenness of different species. So that's alpha diversity. And then beta diversity is sort of looking between samples, between samples. And basically we didn't find association with alpha or beta diversity. And so then we kind of were wondering, okay, maybe there's traits associated with the microbiome that may be associated with autism. We looked at stool consistency, which was an interesting sort of avenue because there are these high rates of gut conditions. We didn't find an association with autism. I do note though that stool consistency is like a snapshot in time measure versus someone having chronic constipation. So perhaps there is a difference there. But again, this dietary signal popped up when we looked at these dietary profiles, and specifically the autistic group consumed less meat. And so these are linear model plots, which I'll show a lot more of later, but basically the response or Y variable is shown as the first here. And then the X kind of variables or the coefficients and their statistics are shown here. And basically no associations except when we come to this dietary PC3, where the unrelated and the sibling group have higher consumption of meat than their counterparts with an autism diagnosis. So to summarize what I've talked about so far, I think it's relatively clear that there have been negligible direct associations between autism and the gut microbiome in our analysis. But interestingly, there was this sort of persisting clues, or this, I don't know, smoke coming from diet. And so then we went back to the diagnostic criteria of autism, one of which are these repetitive and restricted behaviors and interests. And we asked whether those behaviors and interests may manifest as a more restricted diet, or colloquially, we could call it picky eating. And this is actually consistent with the dietetics literature in relation to autism. And we measured restricted diet by just using this alpha diversity measure. So that was pretty simple as well. And we did find an association between autism diagnosis and lower, at least by me, dietary diversity, although there was actually more variance there as well. I mentioned before that there was not association between autism diagnosis and species diversity, and that's shown here. But we wondered whether maybe it's downstream. Maybe species diversity is downstream of dietary diversity. And we did find a nice correlation there, 0.25 Pearson coefficient. So I think that's suggesting to us that yes, it looks like autism, dietary diversity, and then species diversity. And then following this train of thought, we wondered whether species diversity might be associated with stool consistency, and it was. And this is another linear model plot, basically plotting the coefficients for a regression of stool consistency against all these. And basically the only significant term is this species diversity. And specifically what we found is that lower species diversity is associated with runnier, more diarrhea-like stool. Furthermore, these associations were robust to covariates. And so more linear model plots here. But again, Y on the top and the X going down. And we can see finding 1 here, where basically we find an association between dietary diversity and species diversity, and also with diagnosis. For taxonomic diversity, only with dietary diversity. And then for stool consistency, a stronger, at least from a p-value standpoint, association with taxonomic species diversity. Then we wanted to dig a bit deeper into what specifically about autism diagnosis could be driving this restricted diet. And so then we took continuous measures of the autism spectrum from both clinical and genetic scores and looked for their association, and we found significant associations there, even after adjusting for multiple testing. Just as a bit of a side note on what a polygenic score is, basically we have this model of complex traits, and autism is a complex trait. And so the sort of genetic propensity for autism can be captured with these polygenic scores, where basically higher polygenic score means a higher genetic likelihood for that trait. And we basically found an association between genetic predisposition for autism and restricted diet. We also found similar evidence, again, at the genetic and the clinical levels for association between restricted and repetitive traits and restricted diet, even though this sample was smaller for the psychometric measures. And we also found associations with sensory sensitivity or sensory preferences at the marginal level. And this actually makes quite a lot of sense, because if you don't like the taste or the smell or even the sound of a food as you kind of crunch on it, then that's going to restrict your diet in some way. It was also interesting to see that these associations with these autistic traits were stronger for diet than they were with taxonomic species diversity. So here we have dietary, the association with dietary diversity at the top, and then the species diversity at the bottom, and these associations are all stronger for the dietary row. Again, suggesting that autism diagnosis, then diet, then microbiome. We do, of course, caveat that this is a relatively small data set in the grand scheme of things, big for autism, but small overall. And these associations aren't like overpoweringly strong. But overall, I think that on the whole, they do kind of contribute to this model that we propose. And because this is a somewhat contentious issue, I suppose, we ran like a ton of sensitivity analyses. Like each one of these is a sensitivity analysis. So like removing covariates, reducing the age range because age is associated with the microbiome, changing the kind of metagenomics database we used, removing people with antibiotics or probiotics use, only looking at the case control design, only looking at the sibling design, being really careful with rarefaction, looking at different measures of taxonomic alpha diversity, and also looking at dietary quality instead of dietary diversity. That result was actually quite interesting because kids with a more restricted diet also have a poorer quality diet, and that's kind of clinically important as well. In any case, these results all kind of were very consistent with the primary results. So in conclusion, we found that direct microbiome autism relationships are weak, particularly in comparison to other traits like diet, stool consistency, or age. But instead, it looks like autistic traits and preferences restrict diet, and that these dietary preferences in turn are associated with microbiome changes. There are some important implications to this work. Firstly, it tempers the hype around the autism microbiome. And this is really important for families and clinicians who are currently being bombarded with things with little evidence. You know, that's not the best situation to be in. And it's good to provide clarity on that issue. Secondly, I think it also shifts the spotlight in a way from the microbiome to the importance of adequate nutrition for children on the spectrum, just generally speaking, for general development and well-being. We know that that's important. So I think attention needs to be directed there accordingly. And thirdly, beyond autism, I think it also highlights the impact of behavior and diet in terms of inferring microbiome causality in some ways that are perhaps not as systematically looked at as they could be in future. So as I mentioned, this is kind of just one chapter of many other analyses that we're running with this Australian Autism Biobank. So stay tuned for more if you're interested. And this was literally just published last week. So if you're interested in reading more, feel free to have a look. In conclusion, I'd like to thank the massive team that's been involved in this, particularly my amazing supervisors, Jake Bratton and Naomi Ray, who are like my academic parents, everyone involved with the Australian Autism Biobank, the Queensland Twin Adolescent Brain Project, most of all to the families who generously contributed samples and time, and just investment in this work, Microba, computing, and my funding sources, and of course the Gandal Lab for so generously hosting me during my time at UCLA. Thank you so much for your time.